This is Kezia and Abby. We are part of the team investigating gene functions in phages S- and Island 3 as a part of the Seed Genes program at Northwestern College in Orange City, Iowa. We are working with temperate phages Island 3 and S-. The phages have quite a bit of genetic similarity in the left arm of their genomes, but diverge considerably in the right arm, as you can see in this Famirator map. S-'s genome contains 89 genes, only 24 of which have functions assigned to them by bioinformatics. Island 3's genome includes 76 genes, only 17 of which have assigned functions. The genes workflow, as many of you know, has three parts: molecular cloning, phenotypic assays in which we test for the ability of each phage gene to interfere with the growth of the host bacteria, which we call cytotoxicity, and the ability of each phage gene to prevent infection of a host bacteria by another phage, which we call defense. And finally, bacterial-to hybrid analysis used to find interaction partners between host gene products and phage gene products that are cytotoxic or confer defense.
The first step in the molecular cloning process uses PCR to amplify each phage gene from high titer lysates using phage-specific primers. We look for an amplicon size equal to the size of the gene, plus 46 BP, which represents added homology sequences. You can see the kinds of results we get in this gel. In lane 2, S- gene 1's PCR reaction produced a single band of the expected size. And for results like this, we call them purified PCR reaction. In lane 8, we see a band of the expected size and a smaller band. We gel purify results like this. Lane 6 has many bands, none of which represent the expected size for gene 20. This requires troubleshooting. After we purify PCR products, we clone them using isothermal assembly. We transform this into E. coli and plate the reaction on plates with the antibiotic. We pick 3 to 4 can-resistant clones and use PCR to confirm the insert size. In this gel, you can see a representation experiments. In red, all 3 clones of S- gene 23 contain inserts of the expected size, but in yellow, only two of the three clones for S- gene 49 did. We have cloned all 76 of Island 3's genes and nearly half of S- genes. The cloning process produces plasmids that contain our phage gene under the control of a promoter. The reporter gene, M-cherry, is downstream of our phage gene, so when the TEP promoter is active, M-cherry should be expressed along with our phage gene, turning our cells pink. Giving us some assurance that our phage gene is indeed being expressed. With these plasmids constructed, we are ready to assay each gene for cytotoxicity and defense.
These are representative cytotoxicity assays. Each column contains 0 dilutions of M-SNAG electroporated with P extra plasmid containing a phage gene. The plate on the left contains no ATC, so our gene is not expressed. The other 2 plates contain low and high levels of ATC, so our phage gene is expressed. Cytotoxic genes grow in the absence of ATC, but fail to grow when it's present. Non-cytotoxic genes grow similarly in all 3 plates. In the top figure, 3 reps of M-SNAG containing S-21 grow similarly with and without ATC, indicating that gene 21 is not cytotoxic. In the bottom figure, 3 reps of M-SNAG containing S-gene 52 fail to grow in the presence of ATC, indicating that gene 52 is cytotoxic. In the defense assays, M-SNAG containing P extra into what is cloned a phage gene are planted as a lawn of bacteria in top outer. We spot 0 dilutions of phages in each of the 4 columns. If a phage gene confers defense, we expect there to be fewer plaques in the presence of ATC compared to plates where the phage gene is not being expressed, or a lawn containing a controlled plasmid. The top figure shows lawns of M-SNAG containing P extra with S- gene 7. There is little difference in the number of plaques for all 4 phages tested. In the bottom figure, we're testing S- gene 51. This phage gene shows some indication of defense against infection by Island 3, although we'd like to repeat this experiment to be sure. So far, we found 5 cytotoxic genes and 8 genes that may confer defense in this S- genome. We found 15 cytotoxic genes and 3 genes that confer defense in Island 3's genome.
Most of the cytotoxic or defense genes have no known function by bioinformatic analysis. Most of the Island 3 genes cloned into P extra were sequenced by Azenta, and we are part of the process of analyzing those results. You can see a representative result here for part of island 3, gene 13, where 799 bases of our clone gene 13 are identical to the published sequence for this gene. We have a lot of work to keep us busy in the future. And we have many people to thank. Phages and Genes programs, Northwestern College, especially the Department of Biology, Daniel Heller, and all the folks at UMBC, the University of Pittsburgh, and HHMI that make this research possible.